Hi, so I'm Emma Brown, and I'm a research assistant at the Pirbright Institute and in the transmission biology. So my study is going to be on the survival of foot-and-mouth disease in the environment. So sort of following on from what Claire said, so this is based on the data from our transmission experiments. So the study was 4 cattle transmission experiments to study environmental contamination and virus survival. So environmental sampling to estimate the decrease in the environment infectiousness over time and to develop environmental sampling methods for detection for surveillance. So this might look quite familiar from Claire's talk, but I'm purely going to focus on the movement of the donor animals, as this is the animals that were contaminating the rooms. So in room 1, which is bordered in red, was the room that housed the inoculated cattle. So they were inoculated into the tongue with 10 to the 5 of OUKG from 2001. This room was then used for a direct contact challenge for 24 hours before the animals were removed and put into room 2. So in room 1 following this, there was a 24-hour environmental challenge, and then the rooms were left empty and uncleaned for virus survival monitoring. So in room 2, the direct contact donors were then moved in there after their challenge and were left there for 3 days before being removed. And there was another 24-hour environmental challenge in that room before the room was left empty and uncleaned. So as I've already said, the rooms were left empty and uncleaned. If there was a build-up of feces and muck whilst the animals were housed in the rooms, this was swept clean for animal welfare purposes, but it's important to note nothing was washed or disinfected during this time. So the samples collected, in order to sample the environment, we took 2 wall swabs, 5 floor swabs, 5 fecal samples, and 1 food trough. So for the wall swabs, an electrostatic dust cloth was used to wipe a 50 centimetre squared area. So we'd start at one side of the wall and work our way across over the days so that the same area wasn't swabbed more than once. For the feces, we swabbed from 5 different areas. So as you can see from the diagram, it was in each corner and 1 in the middle. So for doing this, we used the 10 by 10 square, which can be seen in the photo. So we'd collect the feces from the square and then wipe the area under the feces. So this wasn't entirely random. Although we stuck to these general areas of the room, we would put the square down where feces was to make sure that we got a sample every day. And then with the food trough, we just wiped down the food trough, again like the wall started at one side and worked our way across the days. So all the samples were collected daily for a week, and all the samples were placed straight into 10 mils of cell culture media immediately, except for the feces, and transported to the lab. So it's important to note that the temperature was roughly about 19 degrees, so about room temperature, and the relative humidity was at 60%. So for the results, in the lab, the samples were tested by qPCR and by virus isolation. So sort of touching a bit on what Claire already showed, PCR was quite consistent throughout the days. We didn't see much drop-off. But I'm mainly going to talk about virus isolation as it is a survival study. So virus isolation was carried out on goat tongue cells. And you can see from the table, this is from room 2. So this is the room with the direct contact donors. And just for reference, day 1 is day 9 of the transmission study. So this was directly after the 24-hour contact challenge, environmental challenge. So we can see that on day 1, we saw a little bit, in the first experiment, we saw a little bit from the floor and the feces, but there was then a complete drop-off from day 2 and for the rest of the study. In study 2, we saw a little bit more up to day 3, but still quite low levels of virus. So unfortunately, due to cross restraints in the first experiment, we only have data from room 1 for the second experiment. So we can see under day 1 is day 5, so this is the first day that the room was empty after the 24-hour challenge. So you can tell that virus was detected on all surfaces, and there was a gradual decrease throughout 6 days and then on day 7 we didn't see anything. So as already mentioned, we altered the experimental plans slightly for experiments 3 and 4, as these were carried out at Parabright rather than at CBI. So as already said, room 1 housed the inoculated cattle and then was taken place for the direct contact challenge. And then room 2 for the direct contact donors, but this time they were moved out of this room before the onset of clinical signs and moved into room 3 where they were left to develop clinical disease. So we have 3 rooms, one with the direct contact donor, one with the inoculated donors, one with the direct contact donors preclinical, and one with the direct contact donors clinical. And to just note that the sampling plan changed slightly, we decided to take 4 wall swabs, like from the floor, using the 10 by 10, and 2 from the food trough. So for the results from room 1, which was the inoculated cattle, day 6 of the transmission experiments is day 1, so that's the first day that the room was empty. We can see that the virus is detected consistently in the second experiment, predominantly from the floor and the feces, whereas it was a little bit more evenly in the first experiment. So in room 2, which is the direct contact donors preclinically, we can see that virus was detected across 6 days, except for day 3 in the third experiment. In experiment 3, there's more of a gradual decrease, whereas in study 4, it gets to day 4 and then there's a complete drop-off. I think I might have flicked past one slide a bit quickly. So that would have been the room from the preclinical room. And this is the clinical room. So virus is detected up to 6 days. I've already said that. Okay, so for the half-lives, the half-lives are calculated for each surface, and they're based on the average of the 4 experiments. So FMD lasted longer in the feces, with a half-life of 6.7 days, and the shortest was on the food trough, with a half-life of 4.2 days. So to conclude, live virus can be detected up to 7 days after the room was left empty. It was, on average, detected highest and longest in the feces, and lowest on the walls and shortest on the food trough. Interestingly, there was not a large difference between the virus titers and survival times between the rooms that were contaminated with preclinical admissions and the rooms contaminated with clinical admissions. So the results of this study will be combined with those of the risk of transmission from an exposure to a contaminated environment, and this will give a better understanding of the effect of disinfection and waiting times before repopulation on the control of FMD outbreaks. So just a couple of thank yous. So everyone at my group at Parabright, so Claire, Simon and Noel, and then everyone who helped out at CBI, Jose, Pedro and Aldo, and everyone, all the animal staff at both institutes for their help. So
thank you very much, Emma. Thank you, Emma. I thought that was a really interesting presentation. It was great. Are there any questions? Yes, Gonzalo has a question here. Get them just here in the middle. Thanks.
Yeah. Well, in the isolation units, they didn't actually have a water trough as such with stagnant water. It was.
CBI, they have basically a hose, a tap that the animals can just take water from. So there was no standing water for us to take samples from.
But yeah, that would be interesting if they did.
Thank you. Michael, I think, has a question here.
Thank you. Since you mentioned like in your last point, the whole disinfection and then repopulation after an outbreak. So at the end
Yeah.
of these, like the data collection over several days, did you just hose down the room and then take another set of swabs, for example?
No, but after the week of virus survival, then the rooms were just left and they were cleaned and disinfected by the animal staff.
And
for,
not swabbed again?
No, we didn't take any swabs after that, but that would be interesting to do, to find. But no, we haven't done that for this study.
You might get an answer you didn't like if you did that. Are there, there's a question here, please. Thank you.
Yeah. Okay. Okay.
Thanks. I think it's a really interesting point, and that's exactly the sort of suggestion I think that these discussion groups are here to bring out. It's certainly something I wasn't aware of. It hadn't even occurred to me. So that's already one extra avenue for us to go down. Are there any other questions for Emma before we move on? No, I think that was a really nice talk. Thank you, Emma.
Thank you.